The cell spends most of its time in interphase during mitosis, which is divided into three parts: the first growth phase, the synthesis phase, and the second growth phase. During the first growth phase, the cell grows and performs many of its intended functions. During the S phase, the DNA duplicates, but is still uncondensed. The nuclear membrane is still intact. In the second growth phase, the cell prepares its organelles that it needs for cell division, including the centrosomes. It duplicates the centrosomes. In animals, each centrosome contains a pair of centrioles. The cell then continues to the mitotic cycle, beginning with prophase. The chromatin condenses and becomes more visible. There are two identical sister chromatids. The nucleoli disappear. The centrosomes move away from each other and form an early mitotic spindle, with microtubules extending from the asters. Then the cell moves on to prometaphase. The nuclear envelope fragments. The microtubules extend and attach to the kinetochores. Some microtubules that don't attach to the kinetochores, called non-kinetochore microtubules, interact with those from the opposite centrosome. The next stage is metaphase. This is the longest stage of mitosis. The centrosomes that make up the mitotic spindle are at opposite poles of the cell. All the chromosomes are lined up at the metaphase plate. Each sister chromatid is attached to the microtubules from opposite poles. Once all of the kinetochores are attached to the spindle fibers, the cell then continues on to anaphase. Anaphase is the shortest stage of mitosis. The chromatids separate from each other and are now known as chromosomes. They are pulled apart by shortening microtubules. The chromosomes move centromere first towards the centrosomes of the elongating cell. Each side of the cell has an equal number of chromosomes. The last phase of mitosis is telophase. In this phase, the daughter nuclei begin to reform as envelopes and nuclei reappear. The genetic contents of each nucleus are identical. Finally, as part of cytokinesis, the cell divides its cytoplasm and splits. In plants, a cell plate forms to separate the cytoplasm as a precursor of the cell wall. In animals, a cleavage furrow pinches the cytoplasm apart like a drawstring. This process leads to two identical daughter cells.